Today and next week, we'll talk about genomics, right? And to talk about genomics, we might as well introduce this idea of omics. This is something that came about modern biology relatively recently. And it comes from technologies that allow us to look at all molecules of a given type within a sample. So just like the sum of genes in an individual or a species we call the genome, the sum of proteins is the proteome, and the study of all of proteins at once in a sample we call proteomics. The sum of all transcripts, RNA transcripts present in a sample is the transcriptome, and the study of that is transcriptomics, and so forth. Lipids, lipidomics, metabolites, metabolomics. So this idea of omics in probably it's fair to say in biology is simply tied to the fact that now newer technologies allow us to look at all of the molecules of a given type within a sample. It used to be that we had to look at things one by one, and now we can look at all of them at once. So when it comes to genomics, the technology that allowed for us to look at all of the genes present in a sample pretty much simultaneously came from advances in DNA sequencing. Now, to understand some of the most modern technologies related to DNA sequencing, it does pay off to take a step back and look at some of the slightly older technologies used for DNA sequencing. And in particular, we're going to be discussing the Sanger method, named after its inventor. So in Sanger sequencing, and by the way, I should preface this by saying that even though it's an older technology, it's very much in use today. If you're working in a research lab and you need to sequence a specific stretch of DNA, chances are that you're going to be doing some Sanger sequencing, by the way. So the technique works as follows. Let's say that we have a template sequence that we would like to find the sequence about, right? We see it here on the screen, but let's pretend for a second that we don't know that sequence. And what we need to do for Sanger sequencing is to start with a primer. It doesn't have to be a super specific primer. Sometimes we do what is called random priming, meaning short sequences that will, one way or another, bind to these template DNAs that we're trying to sequence. And then we do the usual. We mix this primer bound to a template with some DNA polymerase, some dNTPs, and the right buffers, and we let DNA polymerase to go to work and start synthesizing new DNA. So assuming that we have this template sequence, given the sequence that you see on the screen, and we have a primer that, I didn't write it out here, but let's say that it's 5' to 3', meaning the right polarity, the right orientation, you would expect, in the presence of the NTPs, DNA polymerase to start writing the complementary DNA strand to the template. In this case, it would be G, A, T, T, C, G, A, G, C, T, G, A. Right? And then when it gets to the end of that template sequence, we know that DNA polymerase will simply fall off and DNA synthesis ended. So far, nothing new. Now, let's assume for a second that instead of offering the 4 regular dNTPs, we offer DNA polymerase a mixture of 3 dNTPs, in this case dATP, dGTP, and dTTP. And then we offer it, instead of offering dCTP, we offer it a chemically modified CTP that is missing the hydroxyl group at the 3' position of the ribose. If you guys recall, deoxyribonucleotides are called such because they're missing an oxygen atom at the 2' position. But these guys are missing an oxygen atom at the 3' position as well. Instead of being bound to a hydroxyl there, they're bound to simply a hydrogen. So we call these guys di-deoxyribonucleotides. right? They're missing 2 oxygens. Okay? The reason that matters is, if you guys remember, DNA polymerase needs access to that 3' hydroxyl to attach the next nucleotide in the sequence. So if we were to offer this mixture of regular DNTPs for A, G, and T, and then this weird dideoxy CTP, DNA polymerase will start synthesizing and will add a G, an A T, and a T. And when it gets here to the G, it will add that weird dideoxy CTP, but synthesis would end there abruptly because that C doesn't have a 3' hydroxyl that it can offer for DNA polymerase to continue synthesis over that template molecule. So it would sort of end there. I'm going to mark it with an asterisk because that C is missing that hydroxyl. If we do this over and over and over, over many template molecules present in the reaction, or if we were to go through some sort of cyclic amplification by which we detach this new DNA by heating, and then we allow for re-annealing of a new primer, and we let DNA polymerase do it again, DNA polymerase will continue doing over and over and over the same thing. It will extend until it bumps into that dideoxy C and it stops. Bump into that deoxy C and it stops. Right? So all of the fragments that DNA polymerase could generate based on this template, if it had access to only dideoxy CTPs, all of them would be truncated at that C position. That's shown here, right? And the left side of this slide shows what you would expect based on what we just discussed. 3 regular DNTPs for A, G, and T, but that dideoxy CTP. Now, the essence of Sanger sequencing is that we run a trick, a fairly clever trick, if you ask me. What we do is we mix the template with the primers and DNA polymerase, and what we offer DNA polymerase is a mixture that has a lot of D-CTP and then some dideoxy CTP. So the ratio of regular CTP to dideoxy CTP is much in favor of the regular CTP. What does that do? What that does is, and let me check if what that does is some reactions will continue until the DNA polymerase reads the G in the template molecule, and it might happen in some cases that it adds the weird C. Apologies for that, I don't know why I erased it. So it goes all the way there, and then it adds that weird C, and it stops. But because this DNA polymerase has more access to regular CTP, in reality, what's going to happen is more of those reactions will actually read through that G, adding a regular C. Synthesis continues, and for all we know, it might continue all the way to the end. In some cases, the synthesis will continue up to that second G position, and chance might have it that DNA polymerase again picks up one of these dideoxy CTPs, adds it to the growing DNA strand, and now it looks like a G. I meant a C. And then it stops because that C is modified and doesn't have a 3' hydroxyl. So by having just some dideoxy CTP, what we're doing is we're playing the numbers with DNA polymerase. In some cases, DNA polymerase will be able to add a regular C from which synthesis may continue, and in some cases, it may just truncate the elongation reaction. So in other words, what you expect to see is a mixture of fragments of various sizes that stop. If they stop at a C, because that is the one dideoxy nucleotide that we added to this reaction. So they may stop at different positions, but all of those positions are going to be Cs, or the corresponding G in the template. So we haven't really done much yet. I just wanted to introduce this concept because we need that concept to understand what follows. So if we added the regular DNTPs for A, G, and T, and a mixture of regular and dideoxy for C, we're going to get a mixture of fragments, some of which are going to be truncated as Cs. If in 4 different tubes, or in 3 additional tubes, if you will, we change which letter is the one that we have a mixture of regular DNTP to dideoxy. In other words, in this first tube, we have a mixture of regular DATP and some D-dioxy ATP. In this third tube, we have mostly regular GTP, but some D-dioxy GTP and so forth. What we'll have is in each tube, there's going to be a mixture of fragments of various sizes, each of them corresponding at the corresponding letter for which we had one of these weird dideoxynucleotides. Right? So far, we're just looking, in a way, inside of what's going on inside of these tubes. So we haven't really done much in terms of finding out what the sequence of that template was, right? For educational purposes, we know what the sequence is, and I'm explaining to you what's going on. But from a true finding what the sequence is point of view, we haven't really found out anything yet, because all we have are a bunch of test tubes where these reactions are taking place that are generating fragments of various lengths, depending on what letter we had some dideoxy nucleotides for. Here's where the magic comes. We could load this mixture of fragments into a gel for electrophoresis. Not that this detail matters, but instead of using an agarose gel electrophoresis, which tends to have really large pores, we would use a polyacrylamide gel with much smaller pores that allows for resolution of much smaller fragments between them. But the principle is the same. So we load these samples on the wells of that gel. So I didn't draw them before, I'm gonna draw them now. And then we apply the usual, a current, such that you have the negative where the wells are and the positive where DNA would like to go. My question to you, now I hope that you can, as a group, if you will, draw out what you think would happen in terms of where the bands will migrate from each of these tubes depending on their size. So let's see if what you thought might happen kind of matches what we can expect from this. So let me ask you this. Looking at all of the fragments across all of the lanes, in which lane would you expect to see the fragment that traveled the furthest? I hear some louder. G, right? Because I am guessing that you mean this fragment here is the smallest it can be. Only one extra nucleotide other than the primer itself. So that's the smallest fragment across all tubes, and that's the one band that will travel the furthest, right? So there's going to be a band for that somewhere there. What's the second shortest? Somewhere in A here, right? So that fragment from A, so the second shortest band will run ever so slightly slower than that very short from lane G, and it will run in lane A. What's the third shortest? In T, correct. And you get the point, right? So that's exactly what's going to happen in these gels. These bands are going to be traveling a distance that is proportional to their size. And because we're using these special polyacrylamide gels, we can distinguish fragments based on one nucleotide differences in size. All of those agarose electrophoresis gels that we discussed before, those are really good at resolving bands in the hundreds of nucleotides in difference or base pairs in difference. These guys resolve much smaller fragments. Correct. So that is the essence of Sanger sequencing. You generate these reactions that are spontaneously stopped at various positions depending on what letter was D-dioxy. And then you resolve these fragments in a gel, and literally what people did, I know you don't have this slide, but you'll have it later, I'll post it in Canvas. What people literally did for years, actually, is run these gels, then they would kind of draw these lines across the gel, and then they would follow the line from the bottom up, knowing that whichever lane the next band came, that was the letter that corresponded to the next nucleotide in the sequence. In other words, they would start from the bottom up, and they would see that the very first band that they could see was in lane G. So they would say, the first nucleotide after my primer must have been a G. Was the second shortest band the one in lane A? So my second nucleotide must have been A. The third one, T. Then came another T. Then a C, and so forth. Here on the left, you see a real picture of what these gels looked like. And some of these polyacrylamide gels, to allow for longer sequencing reactions, would be kind of tall, sometimes like this. So they, scientists would use special X-ray machines to capture the signal. This was done with radioactive nucleotides. A little crazy, but this is in the old days, right? So I've never run one of these gels myself, not that old, but I've seen the X-rays, and they're fun. They're like huge X-rays, and people would go with a ruler and go from the top up, reading letter by letter. Okay? Cool. So this slide that, again, you don't have right now, but you'll see posted in Canvas later, summarizes the whole process. So using a limited amount of dideoxy NTPs, we can generate fragments of different lengths. Then we run them on a polyacrylamide gel to separate at one nucleotide resolution. We know the nucleotide at the end of each fragment. So then we go from the shortest fragment to the longest. And we can, just looking at which lane a band comes in, we can deduce the sequence. So we can manually reconstruct reading from shortest to longest. Okay? I think I said this a number of times. This is the essence of the Sanger method, and done this way is really all. No one does this. No one does this anymore, right? But the essence of the method was maintained in what is called modern capillary sequencing. As you'll see, the biochemistry is the same. It's just the technology around it changed a little. So at the very beginning of the process, the step is the same as with Sanger sequencing, with classic Sanger sequencing, and that is you need to make many copies of the DNA fragment that you would like to sequence, because that will offer you many template molecules with which to run these partial DNA synthesis reactions, right? So you want many, many template molecules. How do you do that? How do you get many copies of your template DNA? PCR or, So everybody jumps at PCR, and that's fair, because that is the one approach that we would take today to amplify many copies of a given DNA sequence of interest. But there is a method that is actually a little cheaper and easier in some cases to make many, many, many copies of a DNA that you're interested in working with. That's for editing the DNA. Exactly. If you have that DNA in some sort of vector and you can put it in a bacterium, let the bacterium do the work, right? Instead of worrying about designing primers and purchasing the primers, getting them shipped to you, setting up the reaction, using the thermocycler, blah, blah, blah. You just stick a handful, literally a handful of bacterial cells in a liquid culture. You let it grow overnight. You can go home, watch TV, do whatever. The day after, you have billions of bacteria there with your DNA of interest inside of them. And you can extract it relatively easily. And that's it. You have millions of copies of your DNA. All right, so now we have millions of copies, if not more, of our template. And the usual, we denature the template using heat. And then we add only one primer. The key to sequencing is that you're working off of only one primer, right? If you do it with both primers, then you might start getting some sort of PCR going on, and you don't want that. You simply want to read off of only one primer. So you denature your template DNA, you add the one primer on the 3' end of your template sequence. And then, here's the trick, the more modern trick. Instead of dividing the reaction into 4 separate tubes, and to each tube add one of these weird dideoxy letters, what you do is you add a mixture of all 4 of these dideoxy letters, except that they've been chemically modified. Here's where modernity comes in. They've been chemically modified to also fluoresce when you hit them with, I believe it's UV light, right? So these nucleotides are modified chemically in 2 ways. They're missing that hydroxyl group in the 3' carbon of the ribose, and they have a fluorescent molecule attached to them that when you hit them with the right light will fluoresce in a given color. So in this example here, you'll see there is a whole bunch of, I'm going to zoom in, but you can appreciate there is a whole bunch of gray C's, A's, G's, and T's. Those are the regular DNTPs that you need for this reaction to progress to various lengths of that template sequence. But then you spike this mixture with some dideoxy thymidines that happen to fluoresce red light, some dideoxy cytosines that happen to fluoresce blue light, some dideoxy adenines that happen to fluoresce in green, and some dideoxy guanines that have to fluoresce in yellow. Why we do that will become apparent in a second. So, you run the synthesis, the extension from each of these primers, and as usual, you will generate a mixture of partially synthesized fragments, each of which will it will be truncated or finished at a given letter that, because it truncated the reaction because it was missing that hydroxyl, it's also fluorescent. So think of it this way. Each of these fragments will go a certain length, and they will end at a letter that is associated with a given color. So some fragments will end at G, and they will have yellow fluorescence at the end. Some fragments will end in T, and they'll have red fluorescence. Some in A, and will have green fluorescence, and so forth. Okay? All of this is still in a tube. It's all in a mix. So if we were to shine light on this tube, what we would see is a mixture of all of these colors together. Not very useful. We're not really getting any information out of it. So what we do instead is we take the mixture and we load it into a very special kind of gel, again, made of polyacrylamide or some similar polymer, and again, with some sort of well at one end of it. But we call these capillary gels because they're really, really thin, right? We're not going after thick bands that we can see with our naked eye. These are really, really thin gels. They look like capillary tubes, really thin, maybe a millimeter in thickness. But then we do the usual. We subject these samples to an electrophoresis with negative pole where the well is, positive pole on the other end, and these fragments are going to start running through that capillary polyacrylamide gel and separate from one another based on size. Now you have to remember that each of these fragments carries with it a given fluorescence that is given by the identity of that nucleotide where the reaction was truncated. Right? So each of these fragments are stopping in a way at a given position, and the identity of that position will be associated with a given color. And all you do then is subject this gel, you let the bands go through a detector. There is a light source on one end and a detector of fluorescence on the other, and as the fragments are passing through that, there is the detector that says, oh, I see blue fluorescence, oh, I see red fluorescence, oh, I see orange, and so forth. So position by position, the detector will tell you what kind of fluorescence it saw. And that will give rise to these things that I'm showing down here called ferrograms, where at each position you see the corresponding peak of fluorescence that the detector saw. So the first peak here was red, meaning T. The second 2 peaks were blue, meaning C. The fourth was T, and so forth. Right? So this is just a reflection of what is the detector saw as these fragments were passing through. That's Sanger sequencing. That's what most sequencing companies will do when you send out to them your DNA sample of interest that you would like to find out the sequence of. What they will do, what you do is you send them the DNA template that no one knows the sequence of. You will send them the primer that you know will bind to the beginning of that mystery sequence that you're trying to find out. And then what they do is they mix that DNA template and your primer with this mixture of funny nucleotides with different colors. They let the reaction go for a little while, then they load these things onto a capillary tube, and then the capillary tube separates the fragments, and the detector will tell you letter by letter what sequence it saw. Okay? Okay, cool. So I'm going to bring your attention to those numbers at the top of this pherogram. Those correspond to the positions from when the sequencing reaction or when the detector started detecting nucleotides, right? So here this is just a snapshot of sequence somewhere between 120th and 150th nucleotides. And it looks super clean. Each of those peaks of fluorescence are clean, right? Like you see only one color for each peak. And the computer will, with a lot of confidence, will make a call, right? It will say, here there is an A, here there is a C, here there is an A, and so forth. Right? Now, that won't go on forever. I told you that polyacrylamide gels are really good at separating fragments that differ from one another in one nucleotide. So as with anything else in physics, if you're really good at separating things that differ in one unit, by the time they have 800 of those units or 1,000 of those units, you may have a really hard time separating them now. If you're really good at detecting differences in units, you may not be that good at detecting between 1001 and 1002. Does that make sense? So these pherograms tend to break down after, depending on the quality of the sample and the quality of the machine and the maintenance of the machine and a whole bunch of things, they tend to break down after 700 or 800 nucleotides. You start getting these mixed pherograms where there is no single peaks anymore, there is a mixture of peaks. And that is exactly what it sounds like. There is these two fragments that went through the detector together. Why? Because maybe, like I said, one was 812 nucleotides, the other one was 813, and the detector saw them together. So like here, it calls simultaneously a G and an A, I suppose, and when the computer is no longer confident about what the identity of each peak was, it will throw a generic N in your sequence. So see all of those N's that show up? It's basically telling you, look, there was a nucleotide here. I don't know what it was, really. Okay? I bring this up because in the early days of DNA sequencing, people used those long gels, those funny, really long X-rays. Then came capillary sequencing that could do overnight these pretty good sequencing reactions up to 700 or so nucleotides with good quality sequences. But 800 nucleotides sounds like a lot, right? But when you think that an average gene may be 8,000 nucleotides in length with introns and everything, when you think that the genome is 3 times 10 to the 9th, so 3 billion nucleotides, now 800 doesn't sound like much anymore, right? So I wanted to introduce this idea. Capillary sequences, the prep time, you know how I told you that you send these people your template and your primer? For them to mix the funny nucleotides and put it all in the tube and in the incubator, in the machine, the robot, whatever they do, might take about 5 minutes. The DNA synthesis itself takes about a minute. Because DNA polymerases, we've discussed this, they can stage a nucleotide, they can stage up to 1,000 nucleotides per minute, so they can go really fast, right? Then the capillary loading, it's all done by machines. So there is an arm, a robot that goes into the tube, sucks up the liquid, and loads it into the well of the capillary tube. And then, of course, the gel random detection may take a little longer because you need to let these things separate slowly from one another through electrophoresis. So let's say another 30 33, 35 minutes. So it will take you to find out the identity of about 800 nucleotides about 40 minutes. The human genome, as I said just now, is 3 times 10 to the 9 base pairs long. So if you divide the total length of the human genome by what you can get in each reaction and you multiply that by 40 minutes, it would take you approximately 7 years to sequence the human genome. And that is assuming that there is no downtime, that every sequencing reaction is immediately after each other. We don't give these machines a break. We don't give the operators a break. These things are running 24/7, no maintenance, every day of the year, right? When we factor in the fact that obviously nothing can operate at that level, it's more like 10 years, right? My question to you is, does it take 10 years to sequence a human genome? Have you heard these stories about getting your human genome sequenced in about a couple days, right? Yeah, and that is because technology advanced significantly. That's an overstatement, an understatement. It advanced significantly. We'll discuss how that happens. But before we do that, I wanted to bring up something that is just a colorful comment, if you will. Many of you may not have been even born back then, but in the early 90s, Bill Clinton signs an act to sequence the human genome. It was like the Star Wars, but it was the Genome Wars, right? So who was the one to first sequence the human genome? And back then, we didn't have the technologies that we have today that can sequence your genome in 2 days. Back then, we had this technology. The capillary sequences are best, right? So you think, well, these guys were committing to about 10 years to sequence the genome. But the trick they use is all of these that I just said, that it would take about 40 minutes and it will take about 10 years for capillary sequencing to finish a human genome, is if you're using one machine. Let's do the math. If we use 7 machines, it takes more like a little over a year. If you use 7 times 12, so the number of months, it should take a little over a month. And if you do 7 times 52, meaning the number of weeks, it should take, in a year, it should take about a little over a week, right? And that's exactly what happened. In the early 90s, there were these companies that started competing, companies and universities competing for who could sequence the most of the human genome the fastest. And there were these farms for sequencing where you would walk into a warehouse and you would see 50, 100 sequencers all working nonstop at the same time, right? Funny story is that when you go through the math, and with literally way more than 380 machines around the world, like probably closer to the thousands, you would think, okay, so then it took those guys about a month, right? Or a little over a week. It still took them almost 10 years. And the reason it took them almost 10 years to come up with the human genome, so the Human Genome Sequencing Act was signed in 91, I think, and the human genome was published in 2000. So it took about 9 years. The reason for that we'll discuss next week. Anyway, so that was just a short deviation to show you how, and to emphasize something that we'll discuss next week, that sometimes it does help to simply throw everything you have in terms of, in this case, sequencing power at the problem, but even then, it takes a while to do some of these things. With all of that said, today, it literally takes a couple of days to sequence a full human genome, your genome. It's not cheap, but if you have the disposable cash, you can have your genome sequenced fully, right? So how do we do that? What's the technology behind that? So in the reading material for this week, somewhere embedded in that page, there is this video from Illumina. Illumina is the leading company. They have competitors, right? So I'm not claiming that it's the only technology, but it's by far, the leader in the field. So there's a video produced by Illumina itself where they explain to you how their sequencing works. And now we're talking next generation sequencing, which when I say next generation, you have to take that with a grain of salt because this type of sequencing has been around for almost 15, 20 years now. But all things considered, it is still the latest generation of what we call deep sequencing, where you're really going for a lot of sequences all at once. So this is a video there in Canvas within the reading materials page and by itself as a link. But I will go through some of the key steps in a nutshell. So let's imagine that this wiggly black line is a human chromosome, right? It's not your typical metaphasic, highly condensed mitotic chromosome. It's not that X-shaped structure. It's just that wiggly, long wiggly line that is an interphasic human chromosome. What I will describe in the next few slides applies to just a region of that chromosome. But what you'll have to understand in your heads is that what we'll describe for 2 or 3 segments of that chromosome is done simultaneously for all segments of all chromosomes. Okay? So let's imagine for a second that we need to sequence that very short region of that one chromosome that is composed of these 3 subregions, the red, the blue, and the green. With Sanger sequencing, that would have represented 3 separate sequencing reactions, right? 3 separate tubes, 3 separate capillary tubes, 3 separate electrophoresis, 3 separate ferrograms. What next generation sequencing does is it sequences all of these things simultaneously. And it doesn't do 3 at once, it does millions at once. And this is how it works. It all starts by fragmenting your DNA sample into relatively short fragments. Sometimes a couple hundred nucleotides is all you need. So this DNA is fragmented into short fragments, and then with biochemical techniques, let's say the researcher, and let's put it this way, the researcher using Illumina reagents will attach adapters to both ends of each of those fragments. Now, those adapters happen to serve, we will see that in a second, but they happen to serve as primers as well. So for now, let's just imagine that instead of adding primers to a sequence, hoping that they will go and anneal to a given region of your DNA, they're attached to the end of that fragment, right? They're not annealing based on base complementarity. They're being stitched to the end, on both ends of each fragment biochemically, right? There's these enzymes and reagents that will force stitching of these primers to both ends of each fragment. Then what you do is, again, using coarse biochemistry, is attach each of these fragments to what is called a flow cell. And it is called a flow cell because, as we will see in a second, liquids flow through this thing, right? You can think of it as the bedrock of a river, and there is all of these fluids running on top of it. Why that is important will become clear in a second. So for now, let's imagine that these fragments are attached chemically to that platform. I see some of you smiling. I'm like, what are you smiling about? And then I wrote a shitload of primers. Fine. Any questions so far? No? Like that says, you also attach a shitload of primers that are just like the ones that you attach to both ends of these fragments. And they're going to be attached everywhere in this flow cell because you added so much of them. The fragments are going to attach more sparsely in this flow cell because the number of fragments compared to the size of this thing is such that these fragments are going to land relatively separate from one another on the cell. But then you flood this thing with the primers and you attach them all to the surface. Cool. Then what you do is you lower the temperature. You can think of these things, there is liquid on top, right? And you can think of these things as those wiggly inflatable dummies in car lots, right? They're floating around the liquid. But when you lower the temperature in that sort of bending that these fragments do, they may get close to some of the many primers that are surrounding them, right? And when they do, and given that the temperature is lower, they may base pair with those fragments such that now they become attached, not chemically, but through base pairing on their other end to the flow cell. So far, so good? You lower the temperature. You lowered it to, let's say, annealing temperature. So, I don't know, 55 or 60 degrees. That's enough for these things to be moving around. And when they find that primer that is complementary to the primer they have on the other end, they bind through base complementarity and base pairing. So now, here, what you have are typical primers that are bound to a template DNA. Right? If you now, using that stream of liquids flowing through the cell, you send some DNA polymerase and some nucleotides, what do you think is going to happen? DNA polymerase will start synthesizing new DNA. So it will bind to the primer and synthesize over the template. Will bind to the primer and synthesize over the template. Bind to the primer and synthesize over the template. Right? It's like that first round of PCR. What we do then, and that these hash lines are meant to represent that, new DNA molecules that have just been synthesized by the DNA polymerase that you added. What you do at that moment is you heat this flow cell to 95 degrees. What's going to happen? Now all of these things are going to start separating from one another, right? Just like we separated DNA strands in a regular PCR. They were bound to one another because the temperature was cold, but now that it's 95 degrees, they're going to separate and they're going to go back to doing the inflatable dummy thing. Then you do it again. You cool down the sample, right? So you dissociate it, and then you go back to the beginning. You cool down the sample, and now each of these fragments will bend over down to the cell and find a different primer to anneal to. They don't have to necessarily bind to one another. They can, in the random movement, they will find another primer on the platform, and they will bind to it. So we can do it again. And if we do these several cycles, we're essentially doing a PCR on a solid surface. Instead of these things floating around, they're always constantly bound physically to the platform, but there is one free end that is free to find the next primer. We do this several times, and what we achieve is localized amplification of each of the original fragments. Each of those original fragments that had randomly landed somewhere in your cell are now present in several copies. And when I say several copies, I really mean it, thousands probably. We're not going for the millions that we normally go with PCR, but certainly thousands. Because at each step of annealing, there was that bending over of these fragments and binding to the primer that was on the floor, if you will, forming those arced structures. We call this bridged PCR. It's almost like DNA polymerase is walking over one of those old European bridges made of stones, right? And after you've done this several times, you get these clusters. So as the name indicates there, you have some cluster formation in different spots of your cell. Do you know what is on each spot of your cell? Not yet. But what you do know is that each spot in that cell will have many copies of the same fragment. Okay? Now you may appreciate where this is going. In this sense, Sanger's Illumina sequencing is very similar to Sanger sequencing in that you try and start from as many original template molecules as you can, because that's going to give you the ability to see more sequence, right? So there is that initial amplification step, like we did for capillary sequencing with either PCR or using bacteria, right? It's useful to have more starting material than not. So after bridge PCR, you have all of these fragments. Then comes the sequencing itself. And here is where the power of the flow cell comes in. Because what we'll be describing in the next few slides, what I'm about to describe step by step, happens because we have the power to send different things in that flow stream. So we will send a liquid with a given bunch of reagents, let them do their thing, then we flash those out and we send some clear buffer, water, if you will, right? Wash everything off, then send the next set of liquids, let them do their thing, then wash it off, and so forth. And the way you do this, I don't have the visuals for it, but these cells are connected to a series of tubes that are in turn connected to different bottles. And there is a series of pumps and a robot that says, now you should send from this bottle. Now you should send from this one. Now you should send from that one. And on the other end of these cells, there is tubes that will take it out. And in most cases, recycle. Excellent question. I've never built one of these. I never used one of these. My educated guess is absolutely agree with you. What they would probably do is let the water run. It will be dirty at first because it may have some remnants of whatever was there before. And it's like a faucet when you open it after a long while that it comes out brown at first. And then you let it run for a little until it's clear. Then you can use it. So my educated guess is that they let some of that dirty water go, and then they start reusing that water later. Yeah. They're both. They serve as adapters at the beginning when you're attaching them to each fragment and you're using them to anchor these fragments to the platform. But then they serve as primers because when they're attached to the platform by themselves, when these fragments bend over and find them, they will bind to them as primers would to a template sequence in a regular PCR. So they serve both roles. They're adapters/primers. All right, how does the sequencing go? The sequencing goes as follows. The very first thing that comes into these flow cells is a mixture of, again, funny nucleotides. I'll explain in a second how they're funny, and DNA polymerase. These funny nucleotides are funny because they have their nucleotide identity, so they're either an A, a C, a G, or a T. And they have a fluorescent molecule attached to them. And they also have a modified 3' end such that DNA polymerase wouldn't be able to continue the reaction from them. Did I just describe one of those fluorescent dideoxy nucleotides? In a way, but they're not the same. And in fact, these things are kind of proprietary. So I don't know if anyone outside of Illumina knows what they are. They probably do, but my point is that they're not exactly the same as before, but they do the same thing, and that is they will fluoresce when you excite them based on their chemical identity. So A will fluoresce one color, C will fluoresce another color, T will fluoresce another one, and G another. And they also have this ability to stop the reaction because the DNA polymerase cannot continue from their 3' end because the 3' end is chemically modified. So what's going to happen is when DNA polymerase finds these fragments that are now all sticking out, no more arching and binding to their own primers, DNA polymerase will read the letter in the template and will add the corresponding nucleotide to the new DNA strand. So if there was an A in the template, it will add a T that is color-coded here blue. Now that T, as I mentioned, is special. It doesn't allow for any new nucleotides to be added. If it were up to that DNA polymerase would add nucleotides at the rate of about 1,000 a minute. So it would go and fill it up immediately. But it can't because the very first nucleotide that it added stops the reaction. So we know that at each of these positions, there's going to be one nucleotide added, and then the reaction will stop. What we do then is we flow some water or buffer, right? And we get rid of the nucleotides and the DNA polymerase. We probably recycle them. These things are expensive, so the machine will let some of this liquid flow out of the cell and back into the polymerase plus nucleotide bottle, right? We run a lot of buffer to wash off any impurities that may be behind. And then, at that point, since the reaction is stopped or paused, we can take a second to shine the light on these flow cells and use the detectors to see what fluorescence comes from each of these positions. So this fragment here, it happens to be the one color-coded in green here, emits fluorescence for T. So now we know that the position where that particular spot is, where that particular cluster is shining T. The next cluster is shining A. The next cluster is shining G. I apologize, I stole this image from the internet. There is no matching between the color coding of the fragments themselves and the identity of the letters. These colors here are to identify different regions of the DNA that we're trying to sequence. This color code here is for what letter the thing saw in each of these spots. So in one spot it saw a T, in another spot it saw an A, in another spot it saw a G, right? And there's a computer behind all of these that keeps a memory. So now there's this computer that says, well, okay, in this flow cell, in this position here, I have a T, in this position here, I have an A, this position here, I have a G. Cool, now that I know what was the first letter in each of these fragments, I will send in reagents that will unblock the 3' end of that funny special nucleotide. That's why we say that the reaction is not stopped, it's just paused. We send in through the stream a reagent that can unpause the reaction by running a chemical reaction that will re-expose 3' hydroxyl. So now that T became from one that couldn't continue the reaction to one that can. So we unpause it by unveiling the 3' hydroxyl. So we unpause the reaction, and then we send the next round, all controlled by flowing these reagents into the cell. We send the next round of DNA polymerase and the mixture of nucleotides. DNA polymerase comes in, says there is a T there, so it should add an A, and now it can add it because the previous T has a 3' hydroxyl now. So it will add that A, and that A will pause the reaction again, because this A is chemically modified such that the DNA synthesis cannot continue from it. So we wrote the second nucleotide in this sequencing reaction and immediately paused it. We wash all of the nucleotides out and the DNA polymerase like we did before. We wash with buffer several times to get rid of any impurities. And we keep doing this over and over and over again. And at each time, we take a look at which nucleotide was just added. So that computer in the background kept tallies of what was added at each position. It knows that at this position here, the first one was a T, then came an A, then came a G, then came a C, and it just adds those letters to the sequence. At the end of this process, the computer will know that in each position, there was this particular string of sequences, or a string of nucleotides.
It's doing this for millions of sequences at once.
Millions of sequences at once, because these things are associated with microscopic cameras that have the ability to detect micropixels at once, yeah. So millions of sequences at once. These sequences in each of these spots are not super long. Typically, we call those reads, and these reads are in the vicinity of 50 nucleotides. But as we will see next week, if you have millions and millions of 50 nucleotide reads, you can assemble pretty long sequences by simply looking for areas of overlap. But we'll discuss all of that after the break.